Clear? Okay. Is it audible now? Clear? Okay. Hello. Hello. Okay. So wait. Few minutes. You wait. Ah. Now we discuss the fourth paper. So in this paper, here we comprise the four blocks are there. So out of four blocks, here two blocks belongs to the molecular biology. Remaining two blocks belongs to the plant biotechnology. So here first block we have to discuss again here 6 units are there. So each block again comprise the 6 units are there. Okay. So in that 6 units again some different topics also we have to focus. So that first block here, the first unit is the genome and gene organization in higher plants. Okay. And the third unit is the chloroplast and mitochondrial genes. So these chloroplast and mitochondrial gene is the self-replicated genes. So these are the genes are there. They are synthesis those own material. In that third unit is we are to discuss. And the fourth unit is the structure and organization of eukaryotic genes. In the eukaryotic genes, here in the discuss the fourth unit, structure and organization of genes in the eukaryotes. Then fifth one is the gene expression in eukaryotes. This one is a very important. That is the gene expression in the eukaryotes. Then sixth unit is the regulation of gene expression in eukaryote plants. So here this sixth unit is also very important. That is the regulation of genes expression that we are to discuss in the sixth unit. Then coming to the block 2. So block 2 here, that is the block 2. So block 2 here, that is also very important. Block 2. So block 2. Oh. The block 2. So the block 2 is also that is here. In the block 2, this one also here molecular biology topics are there. That is the seventh unit is the restriction endonuclease. Some restriction endonuclease enzymes. Here we are to discuss here and the types of restriction endonuclease. Here enzymes, here different types of endonuclease enzymes also there. We are elaborated in the unit 7. And the unit 8, the modification of enzymes used in the molecular cloning. Modifying the enzymes. Here we are to discuss and molecular cloning law. How we are to use the enzymes in the molecular cloning. Here discussed in the unit 8. And cloning vector. Cloning vectors. So now okay. Right. So and another here can go. And coming to the cloning vectors, here cloning vectors we are to discuss in the unit 9. This is the very important unit, cloning vectors. And 10th unit, we are to discuss the genomic and cDNA library. Genomic and cDNA libraries in the unit 10th. And unit 11th, we are discuss here polymerase chain reaction, PCR. PCR elaborated in the unit 11th, we are discussed here. And 12th unit, now markers, molecular markers. Molecular markers we are to discuss in the unit 12. So coming to the block 2, here block 3. So these block 3 is belongs to the biotechnology. 3 and 4 considered as the biotechnology. Biotechnology topics we are to discuss here. In that 12th unit is the tissue cultures. So plant tissue culture, we are to discuss in vitro morphogenesis, types of tissue cultures and methods of tissue culture. We are to discuss the organ and anther culture, embryo culture, and in vitro morphogenesis. We also discuss here. Okay. In the 11th unit here, anther and pollen ovule culture also. Here, the explant, we are to use the different types of the explants. We are to use for the in the propagation of the tissue culture. And 15th unit here, cryopreservations of plant cells and tissues and germplasm storage. So we are discuss here different cryopreservations. Now we discuss in the unit 15. Now 16th unit is the protoplast culture, somatic hybridization. So in the unit number 16th, now discuss the protoplast culture, somatic hybridization. What is the protoplast and applications also we have to discuss. Protoplast culture applications, somatic hybridization applications we have to discuss here. Then coming to the block 4th, that is the biotechnology. So in this biotechnology here, 4 units are there. These biotechnologies comprise the 4 units. What are that here? 17th unit is the transgenic plants. What is the transgenic plants? Application of transgenic plants we have to discuss. And 18th one is the plant genomics and proteomics. This one is a very important topic in this one. Plant proteomics and genomics. We are discussed in the unit 18th. And unit 19th, some metaboloids, the plant's volumes, plant plant metabolomics we have to discuss. And intellectual property right, IPR, how we have to purchase these IPR and biosafety mechanism and IPR, intellectual property rights, we have to discuss in the unit 10, 20. Okay, clear? So now we discuss here the genome first unit to the first unit. Genome. What is the genomics? And in the first unit, that one is the genome. What is genome? And second unit also we discussed that one, gene organization in higher plants. Discussed here the introduction of this genome. So a genome is a complete set of DNA of an organism. This one, a complete set, complete structure. Genome is a complete structure of DNA of the an organism, including its genes. That means all organisms they have a specific structure of the DNA is there. That one is have the genome. That one we call as the gene. So each genome contain all the information needed to build and maintain the organism and allow to grow and development and do the reproduction system. So what here the each genome means the DNA molecule that is have the all the information about the that particular organism, how they are to develop, how they are to grow, and how they are to synthesis, how they are to doing their reproduction system. All are they are in the uh, they have the particular genome information in that particular DNA. Okay. That is the how. So that they are able to the contain that all the information. In the field of molecular biology and genetics, a genome is the genetic material of organism. You know the genome is nothing but already we discussed that one. The genome is nothing but that is the genetic material means DNA or either RNA. That one we are to consider as a genome in the all the organisms. Okay, so it consists of DNA, either RNA, both the coding and non-coding genes, as well as mitochondrial DNA and chloroplast DNA also. So here the DNA means genome, which is the two types of genetic material is there in individual organisms. They have the DNA type of genomes are there and RNA. Here RNA genome, they have act as a genetic material. Sometimes both are present, so DNA and RNA both are present in that particular organisms, microorganisms or either organisms. So any organism they have sometimes so DNA either material or RNA. Sometimes they are present in the both DNA and RNA. Both are material is there and including the mitochondrial DNA and chloroplast DNA. So these mitochondrial and chloroplast DNA, they are the self-replicated DNA. They are able to their synthesis their own material also means self-replicant DNA which is present. So that's why the mitochondria and chloroplast we are to call as a within the cell. Cell within the cell they have we are to call that one due to the presence of mitochondrial DNA and chloroplast DNA. So another that the study of genome is called as the genomics. So study about the genes means DNA or structure of DNA and genome structure. Nothing but DNA is nothing but a genome. Genome is nothing but DNA, structure of DNA, and what is that molecular characters. All are here. We have to discuss about the that study about the genome is called as a genomics. Okay, a genome is simply the sum total of organisms. That is the DNA is there. Okay. What is history of the genomes? Genome. What is the history here? The term genome was created by 1920 by Hans Winkler. So Hans Winkler is the first introduced by the that genome word. He is the Hans Winkler is the professor in botany at the University of Hamburg in the Germany country. So after 1871, the Friedrich Michel is identified the presence of nuclein. That one we call as the DNA is associated to proteins and cell nucleus. This is there. 1920, the Hans Winkler is the professor of botany at the University of Hamburg, the Germany. Is called the presence of nuclein and associated proteins in the cell nucleus. In 1990, another human genome project is launched at the project aims to sequence in all 3 billion letters of human genome in 15 years. Here we discuss about the human genomes. That is the project is there. And chromosome here 22 is the first human chromosome to be sequenced in this human genome project in 1999. After some day, the full genome sequence of Drosophila melanogaster in 200 years, means 2000 years, they are to discuss. So the mouse is the first mammal to have its full genome sequence completed. In this genome project only, the international mouse genome sequence consumed in 2002. So in the 2002, here we discuss the mouse's complete genome sequences. The mouse genome is the 14% smaller than the human genome, but over 95% of the mouse genome is similar to ours. So this is the some similar information regarding the genome sequence and coding or non-coding character of the organisms like mouse and human beings. Okay, the human gene project, that one is there. Still wait. uh, uh, in the Human Genome Project, which is completed in 2003, and confirmed the human have approximately here 20,000 to 25,000 genes are present in the human beings. Means the genome total genes. So each gene they have their specific characters. So each gene they have specific character is present. The human gene is the sequence to 99.99% that accuracy. Okay. So here, which is ahead means completed in the uh, it. I show to the genes. Wait. I show to the one picture. Mm. This is the here. It's, uh, so this one is the structure of genome, and see here the histone protein and nucleosomes. These are the there, and base pairs here. So this is the genome, means DNA, deoxyribose nucleic acid. That one we call as a DNA, and so this one is the cell repairing process. Here is there. See here. This is the genome structure and remaining all are okay. Now coming to the uh, so that is a gene is completed. That is the wait. So in human genome project, the human genome project is the international scientific research project with the goal of the determining the base pair that make up human DNA and identify the wait and this is the research product to determine the base pair that make up human DNA and identify and mapping all the genes of human genome. So in this human genome project, this is the major project, and to conclude at the how many base pairs are there in human beings and mapping of all genes in that human particular genome. The genome project were provided the complete here 3 billion DNA base pairs. So this research work is allowed to research to being the understand the blueprint of a person, that human beings, particular human beings blueprint. Here we are to discuss in that help of the uh, that human genome project. So the researcher learn more about the function of genes and proteins. This knowledge will have a major impact in the field of medicine, biotechnology, and the life sciences. Now coming to the uh, come to the here genome size. How much of the genome size? Yeah, the genome size, the total amount of DNA contained with the one copy of applied genome. It is the typically measured in the term of mass in picograms of the gram, or in Dalton, or either in the total number of nucleotide base pairs, and usually in the mega bases. This one we are calculate the genome size. In the diploid organisms, the genome size is often used with the term of C volume. A human cell's DNA total about 3 meter in the length. If stretched out, the would from very thin thread about 6 feet long. So the genome size can be measured in the kilo bases or mega bases or picograms. 1 picogram is equal to 798 mega bases are. So what is the largest genome in the world? Here we are discuss in the Japanese flowers named Paris japonica. That is the 149 billion base pairs, making it 150 times larger than the size of human genome. This is the very large size genome is present in the Paris japonica, which is around 149 billion pairs is there. The biggest genome belong to the marbled hungfish, and around 130 billion base pairs are there. In the human cells, most of our body have two copies of the genome, which is 6 billion pairs, and the germ cells only they have one copy of genome, which is made around 3 billion pairs. We know that one. When the sperm and egg cell combine, that is allowed to give the two genomes. So next one is the types of genome. So here types of genome. Now coming to the so types of genomes here. So here so types of genome here, prokaryotic genome that includes the bacteria and virus genomes are there. Another one is the eukaryotic genome that is belongs to the plant genome and animal genome. So here two types of the genomes are there, that is the prokaryote and eukaryote genome. In the prokaryote here bacteria including virus. These here two types we are to discuss, and eukaryotic here plants and animal cells we are to discuss. In the prokaryotic genomes, in the archibacterian, the archibacteria means belong to the prokaryotes. They have the single circular chromosomes are present. The most bacteria also have a single circular chromosome. You know that one. Only the linear structure, thread-like structure is appear on the bacteria, eubacteria, archibacteria. Okay, means they have a specific structure of the nucleus. Nuclear envelope is not present. Only the DNA to looks like a thread or linear like structure is appear, that we call as a prokaryotic organisms. So archibacteria also belongs to the prokaryotes. So most bacteria also have a single circular chromosome. In this have you know that one, the linear chromosome is present, and sometimes multiple chromosome also there. If the DNA is replicated faster than the bacteria cell divide and multiplies, copies of chromosome can be present in the single cell. The most prokaryotes have very little repetitive DNA in their genomes. Some bacteria have extra genetic material, that one we call as the plasmids. In the next class, the another unit is also there. Plasmids act as a vector, means in the gene transformation. When we are to need the specific desirable characters with help of the vectors only, we are to transfer to the another organisms. That topic we have to clear explain in the next topics. So here is the very tiny, very small host cell. If the host cell is present only, they are exhibits. And if they're not available in the host cell, they are in the resting period only. So if the host is available, they are to develop and synthesis their material, and they are involved in the reproduction process and cause of the disease also. So they are the just packages of nucleic acid and protein, that is called as the DNA virus or RNA virus. Some virus maybe have the DNA, or sometimes some viruses they have the RNA or DNA. Sometimes both they have present, depending on the virus. Types of virus here were categorized into these types. So RNA or DNA virus can be either a single strand or double strand genome. Madam.